I like doing things that nobody's done before. I think it's a way to showcase your cleverness. I almost think it's like Apollo 13. You're like, okay, we've got to keep the astronauts alive using these things. This is what we have. And I think it's the same way. Somebody's got a problem that nobody else has solved yet, and here's the tools that you have. One of the studies that I'm working on with a group at Jet Propulsion Lab is evaluating clean room facilities for microbial contamination. So this is part of NASA's what we call planetary protection work, where we're looking to make sure we know what we're sending out into space, particularly if it's going to land on an extraterrestrial body.
I was selected as a crew member to participate in a NASA-supported analog habitat study. Stefan and I will actually be in charge of not only detecting but sequencing microbial presence inside a closed environment. We want to be able to extract nucleic acids using a Reliance instrument, and we're using the Maxwell 16 inside the habitat.
I am a firm believer in automation whenever possible. We didn't want to do everything by hand anymore. My PhD, I was hand making the tubes of beads that would go into bead beating, and it was kind of like a never again. The Maxwell is really one of the easiest instruments to use, and we've also had, over the years, excellent results across a wide range of different specimens.
I've worked with urine, fecal, environmental, soils, from clinical to research samples. It's been all over the place.
Particularly for core facilities, automation is extremely important, both from you want to be able to process a large number of specimens, and minimize the hands-on time, but also from a reproducibility standpoint.
I think this past month, I've ran over 3,000 samples on the Maxwell. So that was fun. It was interesting because at a certain point, it became autopilot. I was processing samples in and out, maybe reaching 4 runs a day.
Maxwell has been kind of our workhorse for a wide range of specimens. You can't guarantee that everybody's going to be working with you for 20 years. And so you want them to, no matter who's in your lab, to be able to run the same thing the same way. We've had, probably 15 years of happy experiences with Promega over the years. So
I think what Promega offers is something simple for complex science.